Good afternoon and good morning. Welcome to this series of webinars that have been organised by the Glyphosate Task Force. My name is Caroline Harris. I'm acting as the moderator for the first two of these sessions. I'm a corporate vice president and principal scientist with Exponent International in the UK. I'm not a toxicologist, but I specialise in pesticide risk assessment for consumer exposure. This afternoon's session is looking at the genotoxic potential of glyphosate. Is glyphosate linked with oxidative stress? We have two outstanding speakers for you this afternoon, Professor David Kirkland and Dr. James Buss, who will assess the scientific arguments at hand and will present on a range of studies and a range of reviews that they have carried out. I'd just like to make it clear that the funding for this series of seminars is being provided by the Glyphosate Task Force, but our speakers have been invited as experts, recognised experts in their field, and are acting as independent specialists. So moving on to our presentations today, I would like to start by introducing Dr. David Kirkland. David was formerly the Vice President of Scientific and Regulatory Consulting at Covance in Harrogate, where he worked for many years for the pharmaceutical industry. In 2009, he left Covance to work as an independent genetic toxicology consultant. David has had many honours over the years, and most recently received the Jim Parry Award for outstanding contributions in the field of environmental mutagenesis in 2015. And this afternoon, David is going to speak to us on the weight of evidence on genotoxicity of glyphosate, its formulations, and UMPA. David, I'll hand over to you.
Thank you, Caroline. Good morning, good afternoon, everyone. I hope you can all hear me. This presentation is on behalf of an expert panel. The names of the other members of the panel are on my first slide. So the views, the opinions I'm going to communicate with you this afternoon are the decisions, the views, the opinions of the panel, and not of any particular individual. And the reason that this panel was convened was to respond to the IARC glyphosate monograph, volume 112, which was published in 2015, which concluded that there was strong evidence supporting that glyphosate can operate through two key characteristics of known human carcinogens: genotoxicity and induction of oxidative stress. And as Dr. Hayes discussed during the webinar on carcinogenicity on Tuesday, and I think Jim has a slide which is also relevant to this in his presentation, these characteristics, genotoxicity and oxidative stress, were used by IARC to support a carcinogenicity classification of probably carcinogenic to humans group 2A. Mechanistic data of this kind has a particular role to play when the carcinogenicity data are limited or insufficient. And even when carcinogenicity data are sufficient, then the genotoxicity is applied in terms of classification. This IARC opinion differed from many previous published regulatory reviews, expert panel reviews, who all came to the conclusion that the proper use of glyphosate and formulations does not pose a genotoxic or carcinogenic carcinogenic hazard or risk. And there are many possible reasons why the IARC conclusion differed from many of the other conclusions, one of which may well be that IARC did not consider a large number of regulatory genetic toxicology studies that have been considered by the other reviews, and including our own panel review. So I'm going to discuss the genotoxicity. There's quite a lot of information to get through. I'm not going to be able to go into detailed data because that would take far too long. But just to consider the studies that are included, how they are assessed, and how they are interpreted is going to take around about 40 minutes. So at the end of my presentation, we will take a pause, and if you have any questions that require clarification of fact, or if you want me to go back to any of my slides to be able to look at them for a little bit longer, then please type in your request during the course of my talk, and then we can take a few moments at the end of it to do exactly that. So the panel was one of 4 panels that were convened by Intertech Scientific and Regulatory Consulting. We conducted an independent review of all of the genotoxicity data, which included a number of studies that were not reviewed by IARC. And we used a rigorous weight of evidence approach to evaluate the results. As will become apparent, it's not clear that IARC used a weight of evidence approach, or even if they did, whether it was a similar weight of evidence approach. And I'll discuss that in a little more detail later on. So inevitably, looking at different data sets, we found some areas of agreement, but we also identified some major differences. If you want the detail, our report is for publication in of critical reviews in toxicology. And as I mentioned, I'm going to discuss the genotoxicity issues, and Jim will talk about oxidative stress in the second presentation. Inevitably, with a substance like glyphosate, which is in widespread use, there will have been extensive testing. And when you have such extensive testing, you end up with a complex data set that contains studies on different endpoints, varied test systems, different exposure methods. And for that reason alone, we believe that it is important to apply a systematic and critical approach to the evaluation. Even the more common test methods often have multiple data entries in the database. And if one thinks simply about calling a study as positive at a significance level of P is 0.05, if you perform 20 studies of the same type on the same chemical and you accept a positive outcome at P is 0.05, one of those 20 studies is going to give you a positive outcome simply by chance. So we're dealing with big and complex data sets that requires a systematic and critical evaluation. So on the next few slides, I'm going to describe the approach that we took to the evaluation of glyphosate genotoxicity. And to begin with human population monitoring studies, of which there were a few, these can offer highly relevant information if they are rigorous, if they have sufficient sample sizes, knowledge of exposure, if they're properly adjusted for confounding factors. But those confounding factors need to consider more than the obvious, more than just age, gender, smoking, alcohol, tobacco, medicines, etc. They should also consider diet, disease status, seasonal variation, physical stress, etc. Nonetheless, we decided to assign a high weight of importance to the measurement of micronuclei in exposed humans. There are many studies using different cell types on glyphosate, and these need to be carefully considered. There are some recommendations in recent years from many scientists that human cells rather than rodent-derived cells should be used, but one also needs to take into account other characteristics such as the p53 status. Cell lines with p53 mutations are typically more susceptible to genetic damage because they don't readily enter apoptosis, but that means that a p53 deficient human cell is just as likely to give the same results as a p53 deficient rodent cell. OECD states that at present, the data do not allow firm recommendations to be made, but suggest it's important to consider p53 status, karyotypic stability, DNA repair capacity, and the origin of the cells when evaluating genotoxicity results. So we believed that it was important in our evaluation to interpret the results of the in vitro mammalian cell tests with caution and to carefully attribute or to consider the weight that they contribute to the overall assessment, taking account of any potential limitations. Particularly with mammalian cells in vitro, there is the opportunity for misleading positive results. We know from several publications that the specificity, the ability to give negative results with non-carcinogens is not as good as we would like, particularly in older studies where the controls were less rigorous than they are now. So we need to carefully consider the impact on the evaluation and interpretation of the results. Misleading responses may be due to non-predictive, or may be non-predictive, that is, they, due to their poor specificity, they give positive responses with non-carcinogens. They may be due to secondary responses, so indirect consequences of high cytotoxicity, high osmolality, low pH, for example, or they may be due to technical deficiencies, such as poor study design, mistakes during the conduct of the test, or inappropriate evaluation. So even more important that a weight of evidence approach should be used. And therefore, individual test methods should be assigned a weight that is consistent with their contribution to the overall evidence. Different types of evidence or evidence categories must be weighted before they are combined into a weight of evidence evaluation. So we applied 4 considerations. Firstly, we argued that different assay types should have different weights, and this is consistent with recent recommendations from the OECD overview of genotoxicity test guideline revisions. That is, that tests measuring mutations and chromosomal damage, which are considered more predictive of cancer hazard, should have greater weight than indicator tests that only measure DNA damage, which may be lethal or may be reversible. Secondly, that the aggregate strength, that is the robustness of the protocols and their reproducibility, and the quality of the evidence in the category should also influence the weight. What that means is that studies conducted to GLP and according to OECD guidelines should have greater weight than studies that lack these attributes. The number of the pieces of evidence within a category also influences the weight, but one or a few divergent responses, so for example, one negative response amongst a host of positive responses, so one or a few divergent responses within a majority of studies with concordant findings should not alter the direction and the strength of the weight of evidence. And finally, tests with a greater ability to extrapolate results to humans should carry greater weight. So data from in vivo animal tests are considered more predictive of potential human hazard and should therefore carry more weight than data from in vitro or non-mammalian cell tests. non-mammalian tests. That is, except for the Ames test. The Ames test as a non-mammalian test is considered to be predictive of potential. So those were the categories of evidence that we applied, and we therefore assigned the studies to one of the following four categories. We had a category of negligible weight, and this is where the endpoint is not linked to any adverse effect relevant to genetic or carcinogenic hazard, and as such, we did not give it any weight in terms of evidence of genotoxicity. Then there was a low weight category. This is where the endpoint is indicative of primary DNA damage, but is not unequivocally linked to mechanisms of tumorigenicity, and where the test system has low specificity. The moderate weight category was for endpoints that are potentially relevant to tumorigenicity, but could be subject to secondary threshold-dependent mechanisms of induction, such as we see with cytotoxic plastogens or with antigens, or where the test system exhibits a high rate of misleading positives with respect to carcinogen prediction or mode of action. And the high weight category was where the endpoint is one that has been demonstrated with a high level of confidence to play a critical role in the process of tumorigenicity. So this is a chart, a table of how we applied those weights to the different kinds of studies that we find with glyphosate. So in the left, that negligible weight was only applied to cystochromatid exchanges in vitro and in vivo. The SCE guidelines have been withdrawn from OECD. We do not understand how SCEs are formed or what are their biological relevance, and so we applied negligible weight to SCE studies. Low weight was given to DNA binding in vitro and in vivo, single-strand breaks such as the COMET assay in vitro, oxidative damage, and DNA repair effects, because all of those are potentially reversible. Moderate weight was given to in vivo strand breakage, in vivo oxidative damage, DNA repair in vivo micronuclei, in vitro chromosomal aberrations, in vitro, and gene mutations in vitro. And high weight was given to the in vivo studies plus the Ames test. So in vivo micronuclei, including human biomonitoring studies, chromosomal aberrations in vivo, gene mutation in vivo, and the Ames test. And this weighting system for these different endpoints in vitro and in vivo is very consistent with the endpoint-specific guidance document of the European Chemicals Agency. So this is not new. This is very much in line with what other experts have considered previously. So we use the considerations I've just described and the weight of evidence approaches discussed above to assess the various genotoxicity results with glyphosate. And as mentioned earlier, whilst we agree with some of the IARC assessments, we disagree on others. So some of those differences may be due to the different approaches. Section 421 of the IARC monograph does not give enough information regarding the strategy employed by IARC in assessing weight of evidence. So we're not sure what they did. We don't know whether the studies were assigned variable weights in accordance with the criteria discussed above. But there is some data that suggests that they were not assigned the variable weights that we just discussed. Because, for example, it does appear from the IARC monograph that in vitro studies in human cells carried more weight than rodent in vivo studies, which is contrary to the grid that I've just shown you. Also, it does not appear that IARC assigned any additional weight to the the relevance of the endpoint in terms of tumor initiation, or additional weight in terms of the quality of the study performance, in vitro versus in vivo, or the reproducibility of the responses. So from our assessment, we agree with IARC that there is sufficient evidence to conclude that glyphosate and glyphosate formulations appear to induce DNA strand breaks and possibly micronuclei in vitro in both mammalian and non-mammalian systems, and cystochromatin exchanges in mammalian systems. But because of the moderate to low weight of these, it is not possible to accurately characterize or classify genotoxic hazard. Which studies should be included? So IARC policies and working group decisions include published papers, but they exclude consideration of additional data from unpublished studies or publicly unavailable government reports. Now, Larry Kier and myself in 2013 published a big review of all of the studies with glyphosate, including a large number of GLP studies. We did not provide the primary GLP reports because it would have taken up too much room, but we provided over 100 pages of supplementary tables in which a lot of detail was included. But IARC considered that this paper, even with its supplementary tables, did not have sufficient details of statistical methods, choice of highest dose tested, or verification of target tissue exposure. So I'm going to deal with each of those issues over the next few slides. Because we believe that the 90 genetic toxicology studies included in that 2013 publication should have been considered, and we believe it makes an impact on the overall conclusion. So although the primary study reports were not available, the detailed data does have an impact, and in fact we believe that the amount of detail that we provided in that 2013 paper actually exceeds the level of detail, the weight of data in most of the published papers that were considered by IARC. There were also some regulatory studies in a 2000 publication by Gary Williams et al. On GBFs and AMPA that were not considered by IARC, and we believe that they should also have been considered. So what's the rationale for arguing that the studies in the 2013 paper should be included? Because we do believe they contain sufficient detail. For example, each study was stated to have been conducted to GLP, and there's no reason to disagree with that. Almost all of the studies were reported to have been conducted in accordance with the relevant OECD test guidelines applicable at the time of the study. If statistical analysis was performed, and basically the only test where it's not routinely performed is the Ames test because one looks for fold increases, but if statistical analysis was performed, this is given as a footnote in all of the supplementary tables in our paper, together with the method used and whether the results were significant. So we don't agree that there was a reason to exclude those studies based on lack of statistical information. In addition, for the Ames tests, the concentrations tested and critical aspects of the method, so for example, plate incorporation or pre-incubation, inducing agent for the S9, final concentration of S9, number of replicate cultures, etc., were detailed in every table. So again, we disagree with IARC's comment. It is clear what top concentrations were used and whether they complied with the maximum concentration as recommended in OECD guidelines, or whether they were defined by toxicity. Almost all of the Ames tests on glyphosate used a top concentration of 5,000 microgram per plate, which is the maximum required. And the tests included all of the required strains, including TA102 or E. coli, in addition to the standard Salmonella strains. For the formulations, the top concentration was actually more variable. Some GBFs could be tested as high as 5,000 microgram per plate, but others were toxic and could only be tested to less than 100 microgram per plate, seemingly due to the presence of the other components of the formulations which are more toxic than glyphosate itself. In the mammalian cell tests, top concentrations generally reached the 500 to 5,000 microgram per mil range, even when prolonged 48-hour treatments were performed, for example, as in the chromosomal aberration studies. So many of the studies exceeded the 10 millimolar limit, which is required under OECD guidelines. Those were studies on glyphosate. There weren't any regulatory mammalian cell tests on formulations. All except one of the regulatory in vivo micronucleus tests on glyphosate achieved a top oral dose of at least 2,000 mg per kg, which is the limit dose according to OECD guidelines. There was one published oral study where the top dose only reached 30 mg per kg, seemingly because of lethality at higher doses, and yet much higher doses were tolerated in other studies using the same acute dosing regimen. So we do not understand why that study only achieved such a low dose. There were several studies that used intraperitoneal injection. Those generally achieved lower top doses because of greater toxicity. So as far as we can tell, all of the regulatory in vivo micronucleus studies on glyphosate met or exceeded the required top dose. So again, we don't understand why IARC rejected consideration of those studies on the basis that they couldn't determine whether an appropriate top dose was used. The regulatory in vivo bone marrow micronucleus and chromosomal aberration studies in our publication were conducted some years ago before it was common practice to measure the presence of the test compound either in the target organ or in the plasma. And because glyphosate is relatively non-toxic, it did not induce toxicity in the bone marrow. So it is correct to ask the question, was the bone marrow exposed? But as IARC state themselves in their monograph, around about 30% of glyphosate administered orally is absorbed and excreted largely unchanged in the urine. So if it's getting into the bloodstream, it's getting into the bone marrow because the bone marrow is a well-perfused tissue. Moreover, there is definitive evidence of absorption and systemic distribution of glyphosate in rodents from various routes of administration in a summary of regulatory toxicokinetic studies in the JMPR report of 2006. There are also a number of publications indicating absorption and systemic distribution of glyphosate by various routes in rats and in mice. So using that information to supplement the in vivo micronucleus and chromosomal aberration tests, there should be no doubt that the bone marrow would have been exposed. So we believe that the 90 additional studies should be included in any overall assessment of glyphosate genotoxicity, and that the exclusion by IARC was not justified, particularly because it was a published paper, and they say they include published papers, and also because the supplementary data covered all of the important information that was required and was actually much more extensive than in many of the papers that IARC did include. And this almost certainly led to a different conclusion and possibly an inaccurate assessment of the genotoxicity of glyphosate, GBS, and AMPAs. What we find is if we look at the IARC studies and then the full collection of studies, we actually see different patterns of positive and negative results, particularly for glyphosate and the formulations, where there were many GLP studies not included by IARC, and particularly for those studies identified as high weight. There were very few GLP studies on AMPA, so in the next few slides, you'll just see data on glyphosate and the formulations. So this is the pattern of results for glyphosate from the IARC evaluation, and I've grouped them according to high weight studies, that is according to the table that I showed you earlier, moderate weight studies and low weight studies. Red is positive, green is negative. So as you can see, for the high weight studies, around about a third were positive. For the moderate weight studies, just over a half, and for the low weight studies, around about 80%. But if we then include all of the 90 studies that IARC did not review, the pattern is quite different. Just look carefully at the left-hand axis. Many, many more studies here, and now we have around 90-odd% of the high weight studies are negative. Moderate weight, more than 50% are negative. The low weight studies are still predominantly positive. But the change in pattern from the IARC profile to the full database profile is really quite stark. So that's for glyphosate. If we then do the same for GBFs, just under a half are positive in high weight studies, and the moderate and low weight studies are all positive. If we now add in the GLP studies, we see again the scale on the left-hand side is much bigger, and over 90% of the high weight studies on GBFs are negative. So this leads us to the following conclusions on the weight of evidence analysis, that test methods identified as low weight produce the highest frequency of positive responses in both of the evaluations. The highest frequencies of positive responses were from endpoints and systems most likely to yield misleading positive results. Indicator tests, P53 deficient rodent cells, or even non-mammalian systems. And this was the case whether it was the IARC evaluation or our recent evaluation. But a minority of high weight studies gave positive results for glyphosate in both evaluations, but the numbers really are quite different. It was 6 out of 15 high weight studies that were positive in the IARC evaluation, but when you add in all of the GLP studies, only 8 out of 92 were positive in the high weight category. So just to summarize in terms of the different endpoints from the full database, so there were 40 Ames tests on glyphosate, GBFs, and AMPA that were not reviewed by IARC, and they were all negative. Glyphosate does not induce gene mutations in mammalian cells in vitro. There aren't any in vitro mammalian cell gene mutation data for GBFs or AMPA, and there's no gene mutation data in vivo. Glyphosate, GBFs, and AMPA do not induce chromosomal damage. They're not plastogenic in vitro. Glyphosate itself is also not plastogenic in vivo. There are some positive in vivo chromosomal aberration studies with formulations, but they're all subject to concerns regarding their reliability or biological relevance, and these are discussed in more detail in our paper. There is limited evidence that glyphosate induces micronuclei in vitro. Now, this could be a reflection of the increased statistical power because more cells are scored in an in vitro micronucleus than in an in vitro chromosomal aberration study. But the absence of chromosomal aberrations suggests that the micronuclei could be due to a threshold-mediated angiogenic effect. And there is strong evidence that glyphosate does not induce micronuclei in vivo. There's no convincing evidence that GBFs or AMPA induce micronuclei in vitro. The overwhelming majority of in vivo micronucleus studies on formulations were negative. The data on AMPA are too conflicting and limited to allow a conclusion on in vivo micronuclei. There is evidence that glyphosate and formulations can induce DNA strand breaks in vitro, but these might be secondary to toxicity since, as we've just seen, they did not lead to chromosome breaks. There's limited evidence of transient DNA strand breakage for glyphosate and formulations in vivo, but we know that for glyphosate at least, these are not associated with DNA adducts, and as mentioned earlier, strand breaks are a low-weight category in any case. There is evidence that glyphosate and AMPA do not induce unscheduled DNA synthesis, DNA repair in hepatocytes. There are some reports of induction of cystochromatid exchanges in vitro and one report of SCE induction in vivo, but as mentioned earlier, these are now considered to have negligible weight and do not contribute to the overall evaluation. There's quite a lot of data on non-mammalian studies. Glyphosate and formulations have been tested in a variety of non-mammalian species other than the Ames test, and they were included in the IARC review. There are 2 positive and 1 negative chromosomal aberration studies with glyphosate in plants, and there are chromosomal aberration studies on GBFs showing predominantly positive results for micronuclei in fish and amphibians. There's a large number of comet assays in fish and other non-mammalian species reported as giving mainly positive results, and there are a large number of positive comet results for formulations in fish and amphibians or reptiles, and 1 positive fish comet study for AMPA. But there are no internationally accepted guidelines for such non-mammalian test systems or any databases of acceptable negative control data or positive control responses, and no results from validation studies indicating whether those assay systems have concordance with carcinogenicity. So it is very difficult to judge what contributions these studies make. OECD guidelines specifically state that use of any non-standard test requires justification along with stringent validation, including establishing robust historical negative and positive control databases. So we do not believe that these studies in non-mammalian systems other than the Ames test should be considered to have significant weight in the overall evaluation. Both major endpoints measured in the majority of non-mammalian tests, so that's the micronucleus and the comet endpoint, in any case may well give positive responses that are secondary to toxic effects. Some of the tests in non-mammalian species involved immersion of the test organism or surface contact with the test material in water. This is not a standard or relevant route of exposure for in vivo mammalian systems, and we just don't know whether it could introduce route-specific, unique, toxic or genotoxic effects. And this is particularly a concern for formulations which are likely to contain surfactants. So we do not consider that the data from a majority of the non-mammalian systems and non-standard tests with glyphosate, GBFs or AMPA have significant weight in the overall genotoxicity evaluation, especially given the large number of standard core studies for gene mutations and chromosomal damage in mammalian systems. Just a couple of slides on human biomonitoring studies. IARC reviewed 3 biomonitoring studies. There was a DNA breakage, a comet assay in humans exposed to GBF spraying that was published by Paziminio et al. In 2007. However, as we've mentioned, the COMET assay is an indicator test and primary DNA damage does not accumulate. So the consequences of the observed DNA breaks remain unknown. It's not clear from the paper which blood cells they were looking at. We don't know whether it was white cells or red cells or all of the cells in the blood. One curious feature of the paper is that the median COMET tail length was exactly 25.0 microns for 20 out of the 21 unexposed control individuals, which seems very curious. More importantly, the sprayers reported signs of clinical toxicity, and the GBF application rate was 20 times higher than recommended. The clinical signs that they reported were consistent with acute intoxication associated with severe exposures. So it is possible that the DNA damage that was recorded was secondary to toxicity. So we feel that this study should be regarded as inconclusive in terms of any evidence for in vivo human genotoxic effects. Pazziamino followed up the same individuals a couple of years later, so that would have been 2009, and then published the results in 2011. And their follow-up was looking for chromosomal changes, and they didn't find any. So that suggests that the DNA strand breaks identified in the earlier study were either repaired or were lethal, because they certainly did not persist as lesions, which could be expressed as chromosomal aberrations in lymphocytes in the follow-up study. There's a 2009 study by Bolognesi et al. That reported a significant but small, transient and inconsistent induction of micronuclei in individuals living in 3 areas where aerial spray application of glyphosate occurred. But the authors concluded that any risk was low. There were no statistically significant increases in the frequency of micronuclei in individuals that actually reported direct exposure to the spray compared to individuals who lived in the spray area but were not present during spraying. This is data from Keith Solomon, and what we've got here are the 2 areas where there was significant spraying, one in red, one in blue. In the left-hand panel are people who lived in those areas but were not exposed. These are micronucleus frequencies, as on the left-hand axis. And then we've got different types of exposure, so spray in the air, spray on the skin, and entering the spray field, and then the consolidated results altogether. And there is no difference. They're all within background noise of one another. So this is the self-reported individuals in terms of spray exposure. So there was no statistically significant difference in post-spray micronuclei between the different categories of self-reported spray exposure, or between no exposure and any self-reported spray exposure. The lack of a significant correlation means, and the inconsistency with application rates indicates that the micronucleus effects cannot be plausibly associated with GBF exposure. So we agree with the authors that based on Bradford Hill criteria, it is not possible to assign causality to the micronucleus increases that were observed. Now, IARC indicated that they had some qualifications of this study in their initial discussion, but they don't repeat those qualifications in their summary, and the unqualified study is given a very high emphasis in the IARC evaluation section as strong evidence for a genotoxic mechanism operating in humans. Our analysis of the data disagrees with that. So based on the above, we concluded that there was little or no reliable evidence produced in these studies that would support a conclusion that GBFs at levels experienced across a broad range of end-user exposures pose any human genotoxic hazard or risk. And finally, just to look at the kinds of characteristics that we would expect to see for a genotoxic carcinogen and what we see for glyphosate, GBFs, and AMPA. So in terms of the profile of responses in various assays, genotoxic carcinogens regularly produce positive results across multiple key endpoints, not just gene mutations, not just chromosomal damage, but positive results across multiple endpoints, both in vitro and in vivo. Glyphosate, GBFs, and AMPA do not do that. Genotoxic carcinogens generally have structural alerts. There are no structural alerts for glyphosate. Genotoxic carcinogens or their breakdown products are typically electrophilic and bind to DNA. There is no evidence that glyphosate or AMPA bind to DNA. Genotoxic carcinogens generally give test results that are highly reproducible, both in vitro and in vivo. With glyphosate and GBFs, we get a lot of conflicting, non-reproducible responses, even in the same test or the same test category. Genotoxic carcinogens routinely give dose-dependent responses over a wide range of concentrations. With glyphosate and GBFs, many of the positive responses do not show dose responses. And genotoxic carcinogens are typically giving positive responses at non-toxic exposures, whereas with glyphosate and GBFs, we are typically finding the small number of positive responses that we do get only in the presence of obvious toxicity. So glyphosate and GBFs do not show the pattern of results that would be typical of a genotoxic carcinogen based on the recommendations of Bolt et al. And Petkoff et al., which is where these categories were derived. So our overall conclusions are that there is substantial evidence, particularly in the Ames tests, not reviewed by IARC, that glyphosate, GBFs, and AMPA do not induce gene mutation, certainly from a direct mechanism, and Jim will discuss oxidative mechanisms in a moment. There is evidence that glyphosate can induce the evidence that glyphosate can produce unstable chromosomal aberrations that might indicate a potential to induce stable chromosome mutations is very limited. It's conflicting, and it's potentially due to secondary mechanisms such as cytotoxicity. The failure of glyphosate and GBFs to induce responses in well-validated test systems with robust experimental protocols that are characteristic of genotoxic carcinogens does not support the conclusion that either glyphosate or GBFs might act via a genotoxic mode of action. Jim is going to discuss oxidative stress in a moment, so I'll skip that one. And there is little or no reliable evidence that GBFs at levels experienced across a broad range of end-user exposures pose any human genotoxic hazard based on the biomonitoring studies. So, in a nutshell, we concluded that the IARC assessment of the classifications regarding strong evidence of genotoxicity and oxidative stress is not supported by the available data. A critical review of the complete data set supports a conclusion that glyphosate, GBS, and AMPA do not pose a genotoxic hazard, and therefore the results should not be considered as support for the classification of glyphosate as a genotoxic carcinogen. So thank you for listening. I apologize that this overran a little bit because of the telephone interruption. I just need to acknowledge Intertech for convening the expert panels and providing excellent organizational and documentary support.
Thank you very much, David. So I've previously introduced Dr. James Buss, who's my colleague from Exponent. And we just wait now for the slides to be handed over. Jim, have you got those? Okay. So these two talks fit very nicely together because Dr. Buss is now going to talk to us about the oxidative stress mode of action in relationship to cancer classification for glyphosate. Thank you, Jim.
Thank you, Caroline. As you've just heard from David, one of the key mode of action considerations, that of genotoxicity, when you look at an overall weight of evidence evaluation of the genotoxicity profile for glyphosate and its formulation, one comes to the conclusion that clearly glyphosate and or its formulations cannot be regarded as genotoxic. The focus of my presentation is going to be on the other additional mode of action consideration, that of oxidative stress, that was used by IARC as part of its overall evaluation of glyphosate, that led it to come to the conclusion that both genotoxicity and evidence of oxidative stress provided supportive evidence in terms of its classification of IARC as a probable human carcinogen. And like what David Kirkland mentioned with respect to the genotoxicity data, even though the oxidative stress data sets are significantly more sparse than that of oxidative stress, nonetheless, I'm still going to focus my presentation on overall high-level perspectives of those data sets relative to what it means in terms of oxidative stress contributing to a possible cancer outcome. However, before getting to those actual data set perspectives, I think it's important for us to understand, first of all, how did IARC come to its focus on oxidative stress as a potential causal mechanism of carcinogenicity of chemicals that fall within its purview of its evaluation. IARC, as many of you know, has long had, appropriately so, and a focus on mode of action information in terms of how it might contribute to the overall assignment of chemicals to the various classification categories that IARC has, ranging from group 1 all the way primarily down to group 3 and a very limited number of compounds that fall in group 4. This particular slide is taken from a slide presented at the Toxicology Forum by Chris Potier, who has participated in a number of glyphosate or a number of IARC reviews in the past, and was originally developed by Vince Cogliano, who at the time was active in terms of engagement as a leader within IARC. What this slide is primarily intended to illustrate is that obviously IARC has two primary considerations that it uses in its classifications of chemicals, that of evidence in experimental animals and evidence in humans. And that's what's illustrated by the different colored blocks that you see on these slides. However, over time, what IARC has realized is with the consideration of mode of action information, as evidenced by the red arrows, there is the possibility that compounds that normally might fall within a given category, let's say by way of example, group 3, that depending on the mode of action database that is available, potentially those compounds actually could be elevated from group 3 into group B, or even transpose up into group 1, or possibly move to group 2A. Additionally, there may be circumstances because of the data sets in animals as well as in humans that a compound might actually reasonably within the IARC criteria fall within a classification such as 2B or 2A. However, in those cases, mode of action information is used as additional reinforcement data to conclude that their decision, in fact, was justified in placing a compound in those two categories. So IARC certainly has realized that mode of action information indeed plays an important role in its overall classification decision. However, IARC, like many other agencies and other institutions reviewing cancer classifications around the world, have increasingly recognized that with the explosion of data associated with mode of action information, and also the increasing complexity of that mode of action data, that clearly there was a need to better understand how that mode of action could be more reasonably condensed into and organized in a way such that the mode of action decisions would be facilitated by improved organization and summarization of that data. And in order to do that, IARC hosted over the last several years a series of workshops, which they subsequently published very recently in a publication authored by Martin Smith in Environmental Health Perspectives in 2016. And over those series of workshops, they evaluated and ultimately came to the conclusion that there are 10 key characteristics associated with human carcinogens. And they're listed here on this slide. And some of those characteristics were just recently highlighted by David in his previous presentation, such as is the chemical electrophilic and metabolically activated to compounds that ultimately could cause genotoxicity. They also recognize that DNA repair and genomic instability are a mode of action which could potentially lead to human carcinogenicity. Others on that list included epigenetic alterations, chronic inflammation, immunosuppressive effects, modulation of receptor-mediated effects, cell immortalization in vitro, and also the ability to induce cell proliferation, cell death, or alter nutrient supply. But more importantly, of course, for the purposes of this discussion today, they also identified oxidative stress as one of those key characteristics. And their intent was to use these 10 key characteristics to help them systematically organize and summarize the data so that it could be better considered for their ultimate downstream mode of action evaluations. Importantly, it should be noted that oxidative stress was not only used in the case of glyphosate, but has actually emerged over the last several IARC reviews as a central mode of action consideration relative to a number of compounds that IARC has reviewed, particularly those of pesticides. By way of example, malathion, DDT, and 2,4-D all had mode of actions associated with oxidative stress that were used in the ultimate classification decisions associated with the IARC reviews. But importantly, the Smith review with respect to oxidative stress made several important caveats relative to the use of oxidative stress. And they recognize certainly that oxidative stress is not unique to cancer outcomes. And it's well known to those involved in mechanistic investigations associated with a number of diseases and other pathological non-cancer states that oxidative stress often emerges as one of the modes of actions that can be associated with non-cancer outcomes. So before examining how IARC actually looks at how they use the 10 key characteristics as it was applied to IARC, it's also important to understand then how IARC developed these characteristics to organize them and summarize them for use in terms of productive analyses of mode of action information. And that's shown here on the next slide. Again, emphasized in the report of Martin Smith and that was associated with the IARC workshop reviews, they emphasized that a review of agents documented and listed as human carcinogens showed a number of characteristics that are shared among many carcinogenic agents. So their fundamental strategy in terms of identifying the 10 key characteristics of human carcinogens was to go into their database for compounds that they had already classified as group 1 carcinogens, and then ask what type of mode of action information do we routinely see associated with those group 1 carcinogens. But it's important to note that those analyses, of course, are only associated with the group 1 chemicals. And then additionally, when you look at the actual data sets that were examined in terms of the individual data, particularly for oxidative stress, as to how do we know whether oxidative stress could be plausibly associated with cancer outcomes, the workshop report cited only 2 high-level reviews of oxidative stress that did not specifically identify how oxidative stress would necessarily be always responsible for cancer outcomes. Importantly, though, it's interesting when you look at the IARC review of how they approached their analysis for oxidative stress and 10 key as being part of the 10 key characteristics, they didn't ask the critical question, how often does oxidative stress, for instance, occur in the lower levels of cancer classifications associated with IARC reviews of chemicals? For instance, group 3, which are compounds generally regarded as having a potentially low hazard for cancer outcomes. And in fact, when you just do a quick cursory overview of chemicals that are found in group 3 classifications for IARC, you find that many of them produce oxidative responses very similar to that for the data set identified for glyphosate. But most importantly, the IARC review also did not include, in terms of their workshop analyses, any analyses of potential counterfactuals that might inform whether oxidative stress can be routinely predicted as being causally associated with cancer outcomes. So by way of example, there are two prototypical pesticides, paraquat and diquat, which are recognized as really the compounds that are representative of high-level oxidant stress inducers. Both of these compounds are well known to be characterized in terms of their ability to undergo cellular redox cycling. In other words, they simply spin off oxygen radicals as part of their intrinsic cellular metabolism. But importantly, because these compounds are pesticides, both of them have been examined in rodent studies for their carcinogenic potential, and neither of them are carcinogens. So here you have a key counterfactual in which compounds which are perhaps the most potent of the chemicals that we know in terms of the oxidative stress community in producing oxidative stress intrinsically within animals, neither of those compounds result in animal carcinogenicity. So that's another key characteristic that was not considered in the overall IARC review. So in terms of further caveats that the Smith IARC review also considered with respect to how you should use the 10 key characteristics, they had several key notations. Certainly, they indicated appropriately so that their emphasis was to identify a method, which is what the 10 key characteristics ultimately emerged as, for organizing and summarizing mechanistic data for the purposes of decision-making and cancer identification, hazard identification. However, they went on to emphasize in this paper that there are several key caveats that need to be considered as you translate the simple organization and summarization of the data into final support level for classification decisions. And they emphasize, first of all, that this approach of summarizing and organizing may be difficult to translate with agents that are both controversial or have limited mechanistic evidence available. And as I'll show you in just a few moments, that certainly appears to be the case with glyphosate. But perhaps most importantly, the Smith publication emphasized that the data that is summarized and collected must be organized to form hypotheses and evaluate evidentiary support for mechanistic events as a function of relevant aspects. And here they specifically emphasize considerations of dose, species, and temporality. And dose particularly was emphasized as being important, and this is certainly recognized for those individuals engaged in mode of action investigations, that those doses must be considered as relevant to real-world human exposures. So with those particular caveats then, the IARC group proceeded then to say, how can we translate the data sets that we have available for glyphosate into an evaluation of the mode of action as it relates to oxidative stress? I should mention before we get to that is there is one additional set of caveats that IARC also mentioned, as is also stressed in a number of publications and reviews of oxidative stress in general. And that is the methodology associated with assessment of oxidative stress, in fact, can be difficult and challenging. So one has to be very cognizant of the methods that were used in terms of characterization of the oxidant stress damage. For example, for instance, the use of COMET assays that are enzyme modified to specifically detect DNA strand breaks associated with oxidative stress are not always necessarily specific with oxidative stress, particularly if the doses or concentrations used in those tests are associated with necrosis or cytotoxicity, which can significantly interfere with the interpretation of those studies relevant to oxidant stress as causality. And importantly, a number of oxidative stress reviews have emphasized that really use of only single oxidant stress biomarkers really is inappropriate, that really it requires a battery of oxidative stress markers to really come to an effective conclusion whether oxidant stress might be related causally as a mode of action in chemical action. So let's turn now to see how did glyphosate actually use the oxidant stress that was available to them, and did they actually follow their own guidance as was published in the publication by Smith. And in fact, here I've just highlighted the data set that was available to IARC, and it does primarily represent the primary data set that is available in the scientific literature. And in fact, when you look at this data set, you can find and readily see that the amount of literature available to evaluate whether glyphosate might induce oxidative stress is indeed very limited. There were only 7 papers that were identified as associating mode of action investigations of oxidant stress in human in vitro cells. There were an additional 7 studies associated with use of oxidant stress in non-human mammalian cells, and then there was a larger cluster of cells or studies associated with non-mammalian species, and I'll come back to in just a few moments why those studies largely should not carry much weight at all in these evaluations. But turning now, first of all, to the IARC consideration of studies that were identified as conducted in human in vitro cells. Again, there were only 7 studies, but importantly, of those 7 studies, 4 of them were conducted using both formulations and or in a mixture. And the oxidant stress that was found in those studies, 4 of the 7 studies, the positive findings were limited only to studies that evaluated formulation or a mixture. Importantly, in these mode of action studies, again, 4 of the human in vitro studies used only a single dose in terms of the mode of action investigation. There are a number of mode of action guidelines and recommendations that have suggested clearly that uses of single doses of mode of action investigations are largely uninformative or associating causality with the mode of action and ultimate toxicity outcome. By way of example, even the Smith publication relative to the identification of the 10 key characteristics cited and referenced a key publication of Cushman et al., in which they did a detailed mode of action evaluation of diethylhexyl phthalate. And in that particular mode of action evaluation, in terms of how they summarized and organized the data, they specifically excluded from consideration of the mode of action studies any study that was only represented by a single dose. So it shows here that studies that have only single doses really should be considered as carrying negligible weight in terms of their overall evaluation. And likewise, the same type of concern applies to studies conducted at only a single time point. And again, oxidative stress reviews, those who are expert in that field of oxidative stress have emphasized that mode of action investigations conducted for oxidative stress using only a single time point, again, are generally uninformative with respect to the causality of oxidative stress relative to its toxicity outcome. And as you can see here, 7 out of the 7 in vitro studies used only a single time point. Another key consideration when you're doing mode of action investigations is that the mode of action should be ideally conducted in the target tissues that have been identified as being of concern. And in the IARC evaluation, they identified both kidney tumors and angiosarcomas in mice as the key target organs of concern. And again, if you accept the IARC conclusion that those are indeed legitimate target organs for carcinogenicity, and I should emphasize here that the webinar that was held previous to this one on this past Tuesday went into depth in terms of how the IARC conclusions relative to the kidney tumors and the hemangiosarcomas should not be regarded as legitimate tumorigenic outcomes associated with carcinogenicity. But regardless, even if you were to accept those endpoints as being real to glyphosate, none of those endpoints were evaluated in any of the human tissues that were evaluated. So they all used tissues that were not associated with either kidney tissues or tissues that could lead to hemangiosarcomas. And then lastly, 7 out of the 7 test methods used only single methods for identification of oxidant stress, and or they used a method that was recognized as having limited value in terms of its ability to detect oxidant stress. So again, in the human in vitro studies, you can see across the board, of the 7 studies that were identified, all of them suffered significant methodological weaknesses associated with their ability to identify oxidant stress as being part of a causal mode of action for a cancer outcome. Now let's turn to the non-human mammalian tests. These are largely rodent studies and or other tissues that were examined. And again, only 7 studies were identified, but importantly, of those 7 studies, 7 of them involved compounds that were tested as the formulation or mixtures. 5 of those studies were limited only to positive findings in the formulation and mixture. So only 2 of them reported positive outcomes that were associated with testing of glyphosate alone. Again, in both cases of single dose or single time point, 6 out of the 7 studies used only a single dose or a single time point. 5 of the 7 studies, again, were examined in non-relevant tissues and or species, i.e. not associated with mice and or rats. And again, all of them had significant methodological deficits in terms of the methods that were used with respect to oxidant stress. In the non-mammalian studies, this slide, the one key point to emphasize here, that is, out of the 19 studies emphasized, 13 of them in this third column here actually came from only a single study, which were conducted in native caught eels using only a single method to evaluate oxidant stress, that of the COMET assay. And those studies actually largely found no effects associated with oxidant stress associated with those investigations. So although those 19 studies are cited as support for oxidative stress, at least 13 of them actually had studies that actually concluded by the authors that there was very limited or no evidence of oxidant stress whatsoever in those studies that were conducted. So what about the key consideration of dose, that being integral to any type of mode of action of investigation? And there is, in fact, a number of key data that can be used with respect to the interpretation of how do the in vitro studies that I just spoke to you about, the 7, how do they actually fit into the context of doses that are used in animal studies used to assess the carcinogenicity of glyphosate. And in fact, as one might expect, there is indeed an excellent set of pharmacokinetic, toxicokinetic data sets that are available for glyphosate conducted in rats. One of those is published by Anodan in Toxicology Letters in 2009. And this is an important observation because the consideration of dose in animal studies actually provides a reference point for the concentrations that are ultimately used in the in vitro studies relative to the doses that are actually induced under conditions of in vivo treatment of animals to glyphosate. And in this particular study, Toxicokinetic by Anodan, they dosed rats orally with glyphosate at 400 milligrams per kilogram by gavage. And importantly, they titrated out the time course of glyphosate toxicokinetic behavior, and they identified a C maximum concentration of 4.6 micrograms per mil. So that is your important reference point concentration to be considered relative to the in vitro studies that I just described to you. And how does that actually inform the interpretation and ultimately of how whether glyphosate might be regarded as a potential inducer of oxidant stress as at the concentrations that they were seeing? Importantly, out of the four out of the 7 in vitro studies actually were tested at LC50 concentration. And as I mentioned to you just a few moments ago, that is important because, in fact, oxidant stress literature has strongly emphasized that interpretation of oxidant stress data in the face of cytotoxicity and or necrotic activity is extremely confounding to its ultimate interpretation, and it has to be regarded with extreme caution. So 4 of those studies were actually conducted at extremely toxic compounds of glyphosate, the LC50. And you'll notice here another important set of observations that come from this. In this particular data, you'll see that studies tested with the formulation, as indicated by the F, actually produced effects at lower concentrations compared to studies that used glyphosate alone. And their concentrations needed to be significantly higher before they produced LC50 responses in those animals. So it indicates that formulations significantly impact the interpretation of oxidant stress data for in vitro toxicology studies. Likewise, that's similarly indicated in this next line, where glyphosate was tested at 900 micrograms per mil as a pure compound, largely was negative. But in that same study, when tested as a formulation, the accident stress data was identified as being present at a dose of 40 micrograms per mil. Again, there are a series of other studies in which glyphosate was identified as producing positive responses, but again, only at a concentration of 580 micrograms per mil. And another study in which it was identified in red blood cells at 42 micrograms per mil. But again, when you look at this overall data set, what does it really tell us relative to the comparator of what can be produced in whole animals as a result of glyphosate dosing? And the test concentrations that were used in these in vitro studies to evaluate oxidant stress all were 9 to 820-fold higher than the Cmax concentrations that are produced in plasma after doses of glyphosate in rats of 400 milligrams per kilogram orally. Now, why is that important? Well, if you were to produce then in order to get the concentrations induced by way of example at an 820-fold higher 820-fold higher in vitro concentration, you would have to multiply the 400 milligram per kilogram oral dose of glyphosate by 820, which would give you obviously an extremely high, literally hundreds of grams per kilogram dose of glyphosate necessary to produce oxidant stress under an in vivo dosing condition. Obviously, that's an absurd dosing condition, and it really indicates further that the in vitro concentrations studied for identification of oxidant stress really were excessively high in terms of informing oxidative stress as a potential causal mechanism for cancer outcomes in humans. Well, what about the in vivo studies? How do they compare to dose? And again, there is a very useful human dose comparator relative to the in vivo studies that were conducted in rats and mice with respect to oxidant stress. Fortunately, there have been a number of high-quality biomonitoring studies that have been conducted with glyphosate. Some of those were summarized in the previous webinar by John Acquavella, but nonetheless, Acquavella himself has published very high-quality biomonitoring studies in which they looked at the total daily dose resulting from exposure to farmers who were actively applying glyphosate, as well as in their spouses and children who were living in close proximity to the active applications of glyphosate. And from those studies, by collecting urine concentrations of glyphosate and comparing those back to total daily doses, you can come up with an estimate that the maximum daily dose that a farmer might have received under those application conditions is 4 micrograms per kilogram per day, while those of the spouses are 0.04 and the children are 0.08 micrograms per kilogram per day. There have been a number of other biomonitoring studies conducted around the world. Some of those have been summarized in a review that I published in 2015, again indicating values very consistent with those of Acquavella, indicating total daily doses that are received in humans as a result of active use of glyphosate ranges from 0.1 to perhaps as high as 5 micrograms per kilogram per day. So how do those dose comparisons compare to doses used in the animal studies? Well, first of all, we see under the animal doses that were evaluated for glyphosate, we find that two of the 7 studies used glyphosate at a concentration of 10 or 300 milligrams per kilogram per day. Importantly, though, this was given by the intraperitoneal route of administration. That's particularly important for glyphosate because glyphosate is only 20 to 30% absorbed orally. So a direct intraperitoneal administration of glyphosate clearly misrepresents the internal systemic dose that can be achieved by an oral dose of glyphosate. Likewise, 2 of the 7 studies, the formulation was evaluated at doses of 50 or 200 milligrams per kilogram, at a single dose, again, given by an intraperitoneal dose. One of the 7 studies actually was a formulation-only study conducted at 50 milligrams per kilogram, but importantly, given by dermal administration. And that likewise is also important because it's well recognized that the dermal absorption of glyphosate is in the range of 1% or less. So again, a study conducted under dermal administration really provides low relevance to ultimate study outcomes. And then lastly, there's 2 studies that were conducted again only with formulations at relatively high gavage dose studies, and a third study that was conducted at 0.38% in drinking water. Importantly, the drinking water biostudy, though, was also critical in that not only did they expose the animals in the drinking water, but once the animals were sacrificed, they actually isolated brain tissue, which is where they were looking for the oxidative stress, and they co-incubated the brain tissue with that same drinking water concentration of 0.38%. So that really represents an extremely exaggerated dose of glyphosate to these animals. And lastly, there was one of the 7 studies that was a mixture of glyphosate, all involving relatively high dose exposures to either glyphosate, cyanide, or dimethylate, all at 15 milligrams, 10 milligrams per kilogram. And again, the route of administration was given intraperitoneally. So again, when you look at these range of test doses, you find that they are 2,500 to 75,000 fold higher than the maximally exposed farmer. And importantly, again, these magnification range, these fold increases range, are based upon direct comparison to external dose administered, not taking into account that the doses actually used in these studies were given either by the intraperitoneal, the dermal, or a gavage drinking water route. So again, the doses used to identify oxidative stress in the in vivo animal studies are excessively high relative to what human exposures are routinely encountered as a result of glyphosate use. Let's turn now to one quick observation with respect to a series of observations relative to how IARC evaluated the non-mammalian studies. It's important to emphasize here that all of the non-mammalian studies were tested as formulation, rendering the interpretive value relative to the mammalian toxicity very uncertain. In other words, all these studies largely involved fish or other aquatic species in which the glyphosate formulation was directly added to the aquatic environment. Again, as I mentioned in one of my earlier slides, 6 of the 19 studies were from the same laboratory, and they resulted in actually negative or equivocal findings using only a single indicator of oxidant stress, which is the enzyme-modified common assay. And it was done again in a species, which again, of course, is certainly not part of a routine species associated with mode of action investigation, a European eel species, which was native caught for the purposes of this particular study. Importantly, out of these 19 studies, only 2 of them evaluated glyphosate only. One of those studies came from the laboratories looking at the European eel, and that particular study was actually negative. The other study showed only a downregulation of the enzyme superoxide dismutase or an upregulation of the enzyme catalase, both of which are of course, antioxidant enzymes. And they looked at, this was shown as expression in zebrafish testis, which of course is a non-target tissue for glyphosate as evaluated in a variety of toxicity studies. So again, the overall evaluation of these non-mammalian target species are very uninformative with respect to mode of action. Lastly, I should mention that one of the studies actually was a mixture study of 8 pesticides done in oysters. So again, that study also is totally uninformative relative to informing oxidant stress as a mode of action for cancer causality. So in conclusion then, what do we really learn about how IARC performed in terms of how they evaluated oxidative stress relative to its ability to inform the mode of action of oxidant stress as a potential cause of cancer in humans? There is really no evidence in the IARC review that they conducted an integrated data analysis using mode of action or any other type of framework evaluation. It certainly appears that all that was done with the oxidative stress data sets that were identified, that they originally said their original purposes was to just summarize and organize. It appears that that is exactly what IARC did. They used those data sets this strategy to summarize and organize the oxidative stress data, and essentially they bin the oxidative stress data sets into the box of oxidative stress. It then appears that they simply looked into that bin and observed in the case of what they saw were 7 papers associated with oxidative stress in vitro in human cells, another 7 papers associated with in vivo treatment of human or mammalian test systems, and another 19 studies that they saw relative to non-mammalian species. But there is no evidence in the IARC review that they went through the necessary integrated data analysis that is required for an effective mode of action analysis. In other words, they failed to consider any of the dose, temporality, coherence, consistency, target organ relevance. None of those key characteristics that are necessary to and absolutely essential to effective mode of action investigation, as is actually even pointed to in their own study emphasized by Smith. None of those evaluations were conducted with respect to glyphosate in the IARC monograph review. So therefore, glyphosate was determined, nonetheless, as a result of them peeking into the bin of oxidative stress and finding the presence of those papers. They classified IARC as having strong evidence of oxidant stress. But importantly, it needs to be emphasized that unlike what David just did a few moments ago with respect to how do you characterize a compound as having low, weak, or moderate strength in terms of its genotoxicity evidence, in terms of defining what those categories actually mean, IARC has provided no criteria for differentiating a weak, moderate, or strong evidence of mode of action relevant to oxidant stress. So it appears that their evaluation is entirely arbitrary. There is no defined criteria whatsoever that would allow them to say the IARC evaluation should lead to strong evidence of oxidant stress. So in conclusion, despite the substantial deficiencies of the oxidant stress analysis that was conducted by IARC, they nonetheless included this data set as providing strong evidence in support of a 2A classification for glyphosate. And I hope you can see from the presentation that I've just described that actually a more reasoned evaluation of the data that was identified as being associated with oxidant stress leads to the very clear conclusion that glyphosate actually has, at best, only weak oxidant evidence of weak oxidant stress. Thank you.
Thank you very much, Jim. We do have some questions that have come in. One specifically for you on your presentation, Jim, and I think it's in relation to your slide 49. And the question is the Cmax of 4.6 micrograms per mil too low for an oral gavage dose of 400 milligrams per kilogram body weight, or is it about right?
It actually is about right. So in other words, there's a number of reasons to believe why that data set correctly is predicting the Cmax. Obviously, they not only identified a Cmax concentration, but they followed the entire time course after an oral gavage scenario of glyphosate. And so it's very clear that their identification of the Cmax time point, in fact, was well done. Secondly, of course, I mentioned that glyphosate is at best only 20 to 30% absorbed orally. And again, these authors tracked how much glyphosate was actually excreted in the feces as a result of this toxicokinetic studies. And they find a significant amount of glyphosate, as expected, was recovered in the feces. So again, the Cmax concentration of 4.6 micrograms per mil is, in fact, very credible with respect to this toxicokinetic study.
Okay. Thank you, Jim. We've got a couple of other questions for you. So why is it important to consider non-carcinogenic oxidative stress in verifying that oxidative stress is a cancer-causing mode of action?
I think the key important there, if I understand the correct question correctly, as I emphasized in my presentation, oxidative stress is clearly not unique to cancer outcomes. There are many disease and pathological states that have been identified through mode of action investigations that have actually relatively strong evidence that oxidative stress is associated with the toxicity outcome. By way of example, I highlighted the pesticide paraquat. Paraquat is well known to cause lung toxicity, and the mode of action of that lung toxicity is well appreciated as being associated with the ability of paraquat to produce excessive oxidative stress as a result of its redox cycling capability. But yet, despite being studied in chronic bioassay, paraquat and its structural analog diquat are not animal carcinogens. So it says one needs to be extremely cautious when one sees oxidative stress, that you actually do need to go through the appropriate framework mode of action investigations in order to establish that the oxidative stress that you're seeing is in fact plausibly associated with the cancer outcome that you're evaluating. And of course, I emphasize again, as I mentioned in one of my slides as well, that endpoint itself is very questionable with respect to glyphosate. More reasoned evaluations of the overall glyphosate animal tumorigenicity data lead a number of expert bodies, regulatory bodies, as well as other expert review panels to come to the conclusion that glyphosate is not an animal carcinogen. So therefore, this is a situation where actually oxidant stress is a mode of action in search of a cancer outcome that has not yet been conclusively identified.
Okay, thank you, Jim. And I think you did answer the next question to some extent, but I'll pose it to you anyway. So most of the studies were carried out on glyphosate alone. What's the value of this when exposure takes place to a glyphosate product?
Yeah, actually, and to correct you, actually, a number of the studies, as I indicated, actually were conducted not with glyphosate alone, but with glyphosate formulation, which really gets at the heart of your question, and that is how do we interpret data with respect to mode of action, or for that matter, even toxicity data that are conducted with mixtures. And again, I think for the purpose of glyphosate, it's very important to come back to the biomonitoring data that we have available for glyphosate that indicates that human exposures to glyphosate are extremely low, even those who are directly involved in the application of glyphosate. And why is that important? There's a number of guidance that's been constructed by a variety of regulatory agencies, one of them the EPA, as well as guidance that's come out of professional societies, such as the Society of Toxicology. And all of those have emphasized that when you are evaluating compounds for toxicity and or mode of action investigations with respect to mixtures such as a formulation or a cluster of pesticides, under which glyphosate certainly has been evaluated for oxidant stress, one has to be extremely cautious in terms of, in fact, the conclusion is that for those compounds that have extremely low human exposures relative to the doses necessary to produce the effects when tested as a mixture, that those mixture findings are uninformative with respect to the ultimate toxicity outcome. In fact, there's a wide body of empirical data that fundamentally show that mixture effects are largely only seen when compounds are tested at the mutual no-L or low-L doses of the individual compounds found in that mixture. But when the doses actually are reduced to the levels of those found in human exposure, such as in the case of glyphosate, where they're literally multiple orders of magnitude lower than the no-L and low-L doses where the toxicity was being seen, that the mixture effects seen under those conditions are uninformative with respect to potential human health risk.
Okay, thank you, Jim. David, are you still on the line?
Yes, I am.
We've got a couple of questions for you, David. So the first one is there's a prevalence of in vitro or comet-like assays in the literature. Can you comment on why this has occurred? And there's a second part of this. What's the importance of applying weighting factors to these findings?
Okay. Why is there a prevalence of them in the literature? Probably because assays like the in vitro micronucleus and the COMET assay have become quite popular in recent years because they are relatively cheap and quick to conduct. And so in terms of getting results and being able to put a publication together, it's much less expensive, much less time consuming than doing mutation experiments, for example, where you would need a lot more in the way of equipment, a lot more plates, a lot more culture media, and a lot more time. So that probably explains the prevalence of those types of studies. Why is it important to apply weighting? Well, in recent years, particularly with the revisions to the OECD guidelines, we've seen a number of revisions that are designed to increase the level of confidence that we have in the outcome of a study. And that's why, and I mentioned it several times in my presentation, the overview of the recent OECD guideline revisions has stressed the need to use test systems that are known to be reliable because we've had many, many years of experience with them. But even with those test systems, that a laboratory needs to demonstrate competence. They need to be able to show that they can get consistent negative responses, consistent positive responses, to have some idea what the background noise looks like in the test system that is being used. So unless you can define the level of confidence that you have in the result, then you're not really in a position to be able to interpret the results in terms of their relevance for humans. So the weighting system that we applied, that others apply, really takes that into account. It's looking at the endpoints that are relevant for tumorigenicity, but it's also looking at the test systems that are considered robust because of many, many years of use, and in which we can have confidence in the results. So the weighting system is really all about how much confidence we have in the test system and the endpoint that is being measured, whether it's in vivo or in vitro, because that informs most critically on how relevant those results are, how reliable those results are in terms of predicting a risk for humans.
Okay, thank you. The next question, actually, you've answered to a certain extent, David, so I'll just pose it anyway. So can you comment on the relevance in weighting human studies in the literature versus the high-weight in vivo studies? I think you presented these in your graphs as part of your presentation, the yes. Red and green bar.
Yeah, I mean, obviously, the most relevant species in which to look for a human hazard would be in human beings. So biomonitoring studies in humans can be very informative, but the studies have to be very, very carefully conducted. The controls that have to be put in place, the measures of exposure, etc., have to be very carefully monitored in order to be able to draw any meaningful conclusions. And the problems with the biomonitoring studies with glyphosate formulation spraying was that there are quite a large number of questions about both of the studies, the Paziminho and the Bolognese studies, that mean that it is very, very difficult to draw any firm conclusions. I mean, even the authors themselves, even Bolognese, came to the conclusion that there was low risk, that the findings they reported were small and transient. And in the Paziminho study, the initial observations were simply for DNA strand breakage. Now, DNA strand breaks may be effectively repaired, or they may be lethal. They may not necessarily lead to a mutational change or even a heritable chromosomal change. And then when they followed up 2 years later in the same individuals, they didn't find any chromosome damage. So if the strand breakage was going to be something which was not repaired, which was permanent, which was passed on from one cell to another, then you would have expected to see chromosomal damage. So although human biomonitoring studies can be very informative, the studies that were conducted on glyphosate spraying really don't give any strong indication that there was any meaningful genotoxic effects in those populations. At least with the in vivo animal studies, we have a couple of advantages. Firstly, we're dealing with a controlled dosing, so we know what the exposures are. We're not dealing with a heterogeneous population. We're dealing with a group of animals that have come from the same breeding batch, so we've got consistency there from one animal to another. and that we can measure endpoints that are not conflicted by confounding factors of other environmental exposures. We can control the environment in which the animals are treated. So because the biomonitoring data really doesn't give us a strong basis for conclusions, we have to then fall back on the in vivo animal data to try to get an impression as to whether or not glyphosate is genotoxic in vivo. So I think that's really how it balances out.
Okay, thank you. Another couple of questions. I think this is probably for both of you. So how can expert groups come up with different conclusions for the same compound?
Shall I take that first, Jim?
Yes, please.
I think just a couple of quick responses to that. I mean, firstly, as I illustrated in my slide set, if different groups are looking at different data sets, then there is every possibility that they could arrive at different conclusions. Now, all of the other regulatory reviews, the reviews by EFSA, by JMPR, by the German BFR, etc., they've looked at the full data set, and their conclusions agree with ours that there is no real evidence of a genotoxic potential with glyphosate. IARC looked at a much smaller data set and came to a different conclusion. So if you're looking at different data sets, you can arrive at different conclusions. If you use different approaches in evaluating the data, you can arrive at different conclusions. So if, for example, you treat every test system and every endpoint as being equal, and you don't apply weight of evidence to the robustness of the test system, the relevance of the endpoint, etc., then again you can arrive at different conclusions. So I think the main answers to why different expert groups can arrive at different conclusions comes from those two aspects.
Okay. Jim, do you want to add anything to that?
And in fact, my answer would be very similar, but with respect to mode of action information, it would certainly indicate that, as you saw from my presentation, I would disagree with the conclusion of IARC with respect to identification of oxidant stress as being plausibly associated with a human cancer outcome, looking at the exact same data set that IARC did. But what was the key difference, and I hope this was picked up on in the presentation that I made, and that is when in any mode of action investigation, and even IARC itself emphasized this point, that it needs to consider, if it's a mode of action study, considerations such as dose, species, temporality, coherence, etc. And even though IARC laid out in their 2016 publications that those were key considerations, in their actual implementation of the mode of action investigation of glyphosate relative to oxidant stress, they actually failed to carry through on their own recommended approach for how mode of action data should be treated. And as you can see from my analyses, when you actually do begin to consider those key elements such as dose or relative target organ sensitivity or species sensitivity, one quickly comes to the conclusion that the mode of action data that was identified for oxidative stress simply is not consistent with being plausibly associated with human cancer outcomes.
Okay, thank you both. The next question kind of follows on from that a little bit. So what's the potential impact of IARC to other regulatory authorities?
Well, certainly.
You want to go, Jim?
Yeah, I'll comment on it. Obviously, the regulatory authorities make their own decisions, again, through their own robust evaluation of the data, again, including mode of action information. So, again, as David outlined just a few moments ago, because of their lengthy experiences and strategies that have been clearly articulated in many written guidance documents, it's not surprising that they perhaps would come to different conclusions relative to the interpretation of data or relative to that achieved by IARC. And of course, it also needs to be emphasized that the purposes of regulatory agencies are entirely different than that of IARC, which very actively states that its reviews are intended only as hazard-based reviews. In other words, they're largely dissociated from any consideration of dose and exposure. Where regulatory agencies are operating under a paradigm that's been in operation in toxicology and risk assessment now for well over 30 years. And that paradigm clearly says that the appropriate way to evaluate chemicals relative to their potential effects in humans is to base the analysis based on risk-based analyses, which of course do have a heavy component of consideration of both dose used in the toxicology studies relative to the exposures that are actually encountered in humans.
Yes, just I would agree entirely with that, Jim. I think just a quick comment to add, I find it quite curious that the IARC monographs have on their front page that they are about the risk of chemicals for carcinogenicity in humans. But as Jim says, they very clearly state in their preamble that they are identifying hazard. And the reason they do this is that whilst at the time that they do the evaluation, human exposures may be very low, and therefore there may not be a risk at that point in time. Who knows what might happen in the future? So they identify a potential hazard. The regulatory agencies will look at those data in terms of what it says in terms of risk.
Okay, thank you. And I've got one.
Go ahead, Carol.
Jim, did you want to come in on that point?
I was just going to emphasize that, again, it comes back to a human exposure consideration. So if you look at the situations where glyphosate is used in the real world, it's as a pesticide, we have ample information that even if glyphosate were suddenly to expand to, many, thousands of acres more of treatment as a result of use of glyphosate, we know that the exposures, even in the worst case situation, those individuals working directly with glyphosate formulation, the actual systemic doses of those individuals is still extremely low. So one can't imagine that there would be any situation where glyphosate might be used in the future where those dose levels would change dramatically. I suppose the only situation where that could happen is if glyphosate was suddenly decided that it might be potentially used as a drug, where you suddenly would start taking it in dramatically higher doses because of potential therapeutic desired outcomes. But in those situations, again, the uses of drugs are highly regulated. Again, so glyphosate in those scenarios would need to go through a complete additional risk assessment relative to its applicability for safety relative to, for drug uses. And it's certainly not anticipated that glyphosate, by way of example, would emerge as a large commodity solvent, by way of example, which would be another scenario in which potential exposures to glyphosate could change in the real world. So when you look at pesticides, and of course glyphosate being one of those compounds in that category, it's hard to imagine how that exposure scenario would change such that the hazard profile as conducted by IARC would be relevant to the real world conditions under which glyphosate is used.
Okay, thank you, Jim. We've got one specific question for David. We're getting very close to the end of our session, but I'll pose this one anyway. So, David, how helpful is it to use threshold mediated to argue about the in vitro micronucleus findings?
Unfortunately, the way the different studies were conducted, we have very little information that would allow us to say that there is clearly a non-DNA target or some other kind of threshold mechanism, for example, based on detoxification or DNA repair, that would argue categorically that there is likely to be a threshold mode of action at work. So at the moment, the data, the studies that were done don't or didn't include those kind of measures, so we don't have that mode of action information. So at the moment, we can only speculate. It is, unless you have a good handle on just what the non-DNA target might be within the cells, and you can demonstrate that there is clearly disruption of that target at the doses that lead to the induction of micronuclei, then you don't really have very solid proof. So at the moment, it's speculative.
Okay, thank you, David. So I'd like to take a final opportunity to thank our really excellent speakers from this afternoon, Professor David Kirkland and Dr. James Buss. Thank you both very much. For the participants, I would just like to remind you that if you have any additional questions, these can be directed via the Glyphosate Task Force website, which is www.glyphosate.eu. And thank you very much for your attendance, and I'll hand back to the web team to close up. Thank you.
Thank you very much for your time today. This ends the webinar.